topic of this presentation is, or the title is Translating the Role of the Complement System in Kidney Disease. My name is Hamid Moradi. I'm a nephrologist, adult nephrologist by training. I'm one of the medical directors at Novartis. And I'm joined by my colleague Julie Koo, who's a pharmacist, who also has done a lot of work in transplant. So the two of us will be doing this presentation. And we're going to start off with Julie starting the talk with giving you some background on the introduction to the complement system overview, and I'll do the complement system overview and the rest of the presentation. Julie, I'll turn it over to you.
Thank you. So, welcome to the section. So complement system plays a central pillar of the innate immunity and plays a role in adaptive immune system. And probably some of the audience you are all the immune systems experts. What about the dysregulation of the complement system? So it may have a potential to cause autoimmune, inflammatory, degenerative, hematological and ischemic disorders, and also including the glomerular disorders. So recently, the dysregulated complement system has gained recognition to be a driver for the pathogenesis and progression of several types of primary glomerular nephritis, known collectively as CMKD. CMKD stands for complement-mediated kidney disease. So I'll mention a couple of them, such as IgA nephropathy, complement 3 glomerulopathy, the immune complex-mediated MPGN, atypical HUS, and lupus nephritis. So the current KDIGO consensus paper has suggested the complement play a crucial role in the pathogenesis and in some primary rare disease with the membranous proliferative glomerular nephritis, MPGN pattern of injury. And the extent of the contribution from complement system, so as you can see from this graph, the condition listed on the left-hand side, including atypical HUS, C3 glomerulopathy, and primary immune complex MPGN, complement dysregulation plays a primary role, versus to the far right end, such as diabetic nephropathy, FSGS, complement dysregulation may play as a secondary driver for this condition. So we may need to ask, what about the potential impact of the complement inhibition? So as I mentioned, all this, the rare condition, atypical HUS, IC-MPGN, and also the C3 glomerulopathy, they all share the similar pattern of the injury. So it's a membranous proliferative glomerular nephritis. So the MPGN pattern of injury is characterized by, you guys are the experts, the nephrologists, increase in the intraglomerular cells, mesangial hypercellularity, endocapillary proliferation, and diffuse thickening of glomerular capillary walls. So you can see under the light microscopy for this MPGN pattern, but with the immunofluorescence and staining technique, and also the electron microscopy that can help further identify the underlying disease etiology of this MPGN pattern of disease. So in most cases of the MPGN, lesion is results from the immunoglobulin, as you can see on the left column, driven by immunoglobulin or the complement deposition. And also for the immune complex mediated secondary to the autoimmune diseases or infection, or if that's immunoglobulin as the primary deposition on the MPGN pattern, it might be from the immunoglobulin-mediated conditions such as monoclonal gammopathy. So the one listed in the middle, if the lesion is primarily complement deposition, specifically for C3, those are considered to be complement-mediated condition, including both C3 and C4 glomerulopathy. And furthermore, through the electron microscopy, that can further divide it into two subgroups, the dense deposit disease and glomerular nephritis. And then, of course, there's another subcategory. If the lesion doesn't present with any immunoglobulin or complement C3, those are the other potential etiology, including HUS or the TMA associated condition. So as I mentioned, the KDIGO has been recognized. Some of the primary the rare disease, such as C3G, immune complex MPGN, and atypical HUS, the complement dysregulation plays a primary role. And also the diagnosis is somehow complex. And also the progression is really severe. As you can see, the incidence listed here, they are anywhere from 1 to up to 4 per million per year of the incidence in the US. And due to the rarity of the disease and also the delay on diagnosis, so patients' outcome may not be so great. So you can see the progression. So with a C3G, IC-MPGN, and atypical HUS, so you can see the anywhere from 30 to up to 70% of patients will progress into the kidney failure within 3 to 15 years. And if these patients are lucky enough to receive kidney transplant, so the recurrence in those, for example, in the C3G condition, the recurrence in histological recurrence is up to 90%. And then you may ask, so what? So you can look at the recurrence of C3G can lead to the allograft loss, so anywhere from 50% to 70%. And so the other striking documentation, this recurrence can occur almost immediately post-transplant. So now I'll turn on to Dr. Moradi.
Thank you very much, Julie. Now, with that background on C3G and complement-mediated kidney diseases, let's talk about the complement system. And I think this picture kind of takes you back to first year of med school or the very first physiology or immunology class that you had. The complement system at first glance seems scary, lots of arrows and letters and numbers. But if we were to simplify this and break it up into three parts, the first part of the complement system is activation. And as you can see, you can activate the complement system via 3 different pathways: the classical pathway, lectin, and alternative pathway. But regardless of how you activate the system, all 3 pathways merge on formation of this enzyme called C3 convertase. It's a serine protease, and as the name would suggest, it cleaves C3 into its components, C3a and C3b. Now, Once you have C3b formed, there comes the amplification loop. And as you can see, the amplification loop of the complement system lies in the alternative pathway. That's one of the things that makes the alternative pathway unique, and we'll be talking about this more. And so the C3b can feed back into the C3 convertase of the alternative pathway and create more C3 convertase. As you can see in this side, the C3 convertase of the other two pathways doesn't have the C3b component, so they can't amplify the signals. But the alternative pathway C3 convertase does have this. So you have amplification of the signal, and depending on who you read, some may say that the majority of the overall activity of the complement system comes from the alternative pathway because of this amplification loop. So it will take signals from the other two pathways and it will amplify it too. Once you have amplification and you have more C3b formed, C3b is also an integral component of an enzyme called C5 convertase. C5 convertase then goes on to cleave C5 to C5a and C5b. C5b goes on to form the membrane attack complex that we all have heard about. It causes pores into cells, or it could be bacterial cells, or even it could be cells, our cells. And so that's where the terminal sequence is. But these other fragments, C5a and C3a, also have their own functions. They're anaphylatoxins. They have downstream effects as well. So as I mentioned before, the three pathways for activating the complement system are the classical pathway. The classical pathway is usually triggered by antigen-antibody immune complex deposition. Lectin pathway is activated typically by mannose-rich bacterial surfaces or carbohydrate-rich surfaces, and it doesn't necessarily have to be bacteria. It could be damaged cells. And then the alternative pathway, two things make it unique. One is that it's always on at a basal level through this process called the tick-over process. So in all of us, the alternative pathway of complement right now is at work at a basal level. And then the other part that, as I mentioned before, that makes it unique is that the amplification loop of the entire system lies in the alternative pathway of complement. And so why is it that we have right now alternative pathway activity, but our complement system is not dysregulated? It's because there are regulators and then there are promoters of activity. The regulators of activity put the brake on the system, and we'll talk about some of those, things like factor H, factor I, and the promoters promote activity of the system. Properdin will be one of them, and that's this little dot over here that stabilizes C3 convertase. So C3b is also an opsonin, and what that means is that it tags things for destruction. So it tags, for example, red cells. It can tag them for destruction by phagocytes. So that's another function of C3b. And then we also talked about the fact that it's a major component of C5 convertase, which then activates the terminal pathway, MAC formation, and then we also talked about anaphylatoxin activity of C3a and C5a. So every component in this picture has a role. And so how does the complement system become dysregulated, and what are the implications for the kidneys? So the complement system dysregulation can be caused by genetic abnormalities, or it could be acquired, meaning you could have autoantibodies, for instance, that stabilize your C3 convertase. And regardless of how you get there, the kidneys are particularly vulnerable to complement activation and damage because of complement activation due to several reasons. One is that the glomerular structure, as you all know, the kidneys see a large volume of blood. 1/3 of the cardiac output goes to the kidneys. They're seeing a high concentration of complement proteins and also immune complexes. And you also get immune complex deposition in the kidneys. It's not uncommon for us to see that on biopsies. While most of the complement proteins are made by the liver cells, by hepatocytes, some of the complement proteins can also be made by kidney cells. So C3, for instance, up to 10% of C3 can actually be made by the kidneys. Some of the studies have shown this. And some parts of the kidney lack those control mechanisms, the regulators, the lack the regulators of complement system. For example, the glomerular basement membrane doesn't have the surface proteins that typically regulates the complement system. So because of that, the kidneys are especially prone or vulnerable to damage by the complement system. So going back to this picture, it is interesting that even though dysregulation of the overall system is involved in kidney disease, it can manifest itself depending on what is actually missing in different conditions. For example, a dysregulated alternative pathway activity can cause C3G or immune complex MPGN, or it can cause atypical HUS. And this talks about the downstream effectors of the system, C3A and C5A, and also the membrane attack complex. And the membrane attack complex also has a soluble component that can also damage kidney cells. So this goes into the details of, what do each component do? And some of this data is from in vitro, from cell culture, from animal data. But there's a slew of events that take place once you form these fragments, a lot of them involved in inflammation, and they're involved in promoting inflammation, activating mesangial cells, damaging tubular cells. What results is you get inflammation, glomerular injury, and scarring, and it manifests as hematuria and proteinuria, and ultimately it can result in kidney failure. So the gold standard for diagnosing complement-mediated kidney disease is biopsy. And I'm not going to preach to choir. You know this better than me. This is how we examine kidney tissue. Our pathology colleagues use light microscopy, immunofluorescence staining, and electron microscopy. And while the complement system laboratory assays or laboratory evaluations can have a role, it can give you some information, the actual diagnosis will come from biopsy. Some of the ways that you can assess the complement system through the laboratory assays or tests is you can have a functional assessment. You can also measure serum levels of different complement components, the different complement proteins that are circulating or in the urine. And you can also do genetic testing. A lot of these tests will require specialized tests and specialized laboratories that are experts in this. Now, on immunofluorescence, you can have clues as to which pathway of the complement system is being activated. Although not definitively, it just provides you with some clues. For instance, if on immunofluorescence staining you see C3, C4D, C1q, that is more consistent with activation of the classical complement pathway. Whereas if you're missing C1q, and you have C4D and C3, it's more likely that the lectin pathway was activated. If you're missing C4D and C1q, then it's highly likely that it was the alternative pathway that was activated. These are just hints. It doesn't definitively tell you this, and I think we just went over that right now, so I won't repeat myself here, and then we'll move on. Here are some of the tests that can be done at some of the commercial labs. Most labs can measure C3C4, serum C3C4, and a lot of labs can measure CH50, but measuring the functional assay for assessing, for example, the function of the alternative pathway, not a lot of labs can do this. The nephritic factors also and genetic testing are typically done in specialized labs. But these are some of the examples of how you can evaluate the complement system through laboratory testing. So now let's talk about C3G. Let's go back to C3G again. It's a complex, rare, complement-mediated kidney disease that's the hallmark of C3G, as the name would suggest, is deposition of C3 in renal tissue. There are two subtypes, C3 glomerulonephritis, and it gets really confusing because you say C3G is the umbrella term, and then C3GN is the subtype, is C3 glomerulonephritis, and then you also have dense deposit disease. There are some differences between the two, and we'll talk about that. It is a rare disease, so the incidence is 2 to 3 per million in the United States. It affects individuals of all ages. It could be, we could start in childhood and go all the way into adulthood. And the incidence is similar in men and women. It presents typically with hematuria and proteinuria. It can also present with nephrotic syndrome, but as you can see, this is not a distinguishing feature. A lot of glomerulonephritis present with these same characteristics, or the presentation is very similar to a lot of other glomerulonephritis. You can have extrarenal manifestation, so acquired partial lipodystrophy is something that can be seen in 5 to 17% of patients. You can also have ocular complications, drusen deposition, the stuff that you see in age-related macular degeneration. You can have it in a minority of C3G patients, so that can also be seen. It's typically, it could be stable for years. but show up with persistent proteinuria, or it could have rapid progression. So it's highly variable how it can present and how it can go forward. But the pathophysiology of it involves the dysregulation of the alternative pathway of complement. And as I mentioned before, we talked about this, the tick-over process and the amplification loop. And here are some of the regulators that we were speaking of. So if you have mutations in factor H or factor I, or you have an antibody that stabilizes properdin and you can't break down properdin, again, you get dysregulation of this alternative pathway activation, overactivation of the alternative pathway, which can lead to C3G. The dysregulation of the alternative pathway is present in more than 90% of the patients, but genetic abnormalities are present in around 25%. So just because someone doesn't have a genetic abnormality doesn't mean that they don't have C3G. And so diagnosis again, initial presentation would prompt you to do a kidney biopsy, and then on biopsy you would have either sole C3 deposits or dominant C3 deposits, meaning more than 2 orders of magnitude greater than any other immune reactant. And some of the immune staining, some of the immunofluorescence staining can be helpful in directing you to the alternative pathway, as I mentioned before. Now, you can distinguish between C3GN and dense deposit disease through electron microscopy. Dense deposit disease has this ribbon-like deposition in the glomerular basement membrane that you don't see with C3GN. And that's kind of the hallmark feature of dense deposit disease and how you diagnose someone with dense deposit disease as opposed to C3GN. And again, this, to me, just highlights the fact that you don't have to have low C3 to have C3G. Not typically, about 50% of patients will have low C3 levels. You may also have low activity in the alternative pathway of complement, low factor H, low factor B. A lot of this is due to consumption. Because the system is so revved up, it's using everything up, so then you get low plasma levels. But none of them are pathognomonic for C3G. These are some of the things that you can see. And you test for them through specialized labs. C3G does have a differential diagnosis. Even if you have done everything and the biopsy shows C3G, there are other conditions that can present in a similar way. And so if you look at the clinical presentation, it's very similar across the board between C3G post-infectious GN and IC-MPGN. And there are some mild differences. So maybe you see more nephrotic syndrome with C3G. Typically with C3G, you don't see any other, or the immunoglobulins are low, there's low IgG, whereas with post-infectious GN and IC-MPGN, you see more IgG deposition, and those could be some of the differences. And then at the end, again, you can have normal or low C3. That doesn't rule out, having normal C3 doesn't rule out C3G. With IC-MPGN, you may see C4 being decreased, again, because you have these immune complexes that can activate the classical pathway as well. But again, this is a differential. I think for post-infectious GN, typically what people say is you have to wait for 3 months. If things don't normalize, if things don't go back to normal, then you suspect an alternative pathway dysregulation and you do the workup or the testing for the complement pathway testing and the genetic testing as well. Prognosis, as Julie mentioned, is somewhat poor. Spontaneous remission is uncommon. 30 to 50% of patients develop kidney failure within 10 years. And this is another UK RaDaR study from England. They have a RaDaR MPGN analysis, and they've said that the annual eGFR decline was around 5 ml per minute per year in patients with C3G. There are limitations to this study, but this is just one data point that was recently published. And dense deposit disease is more likely than C3GN to progress to kidney failure, but C3GN is more likely to recur in the transplanted kidney, and in both of them result in graft loss. These are all based on studies with really small numbers, so I think in the end the differences might not be huge, but maybe some differences between the two conditions. So in summary, and I'm sorry that I went fast, the complement system is comprised of the 3 pathways, classical, lectin, and alternative pathway, and is a key part of the immune system. And more we're recognizing that it plays a role in glomerular injury. And this regulation of the system can be caused by genetic mutations, or it can be caused by autoantibodies, and it contributes to pathophysiology and progression of several types of primary glomerulonephritides. We talked about C3G here, but it can also play a role in immune complex membranoproliferative glomerulonephritis. A kidney biopsy is the gold standard for diagnostic evaluation. Complement testing might help, but it doesn't give you the diagnosis. You need a kidney biopsy. And there may be some differences on biopsy findings when you do compare IC-MPGN to C3G. So with that said, I'm going to end this talk and allow you to ask any questions you would like. The QR code, if you can scan it again and let us know how we did, we would highly appreciate it. Another resource that we have available, the website we have up there called Renal Halls of Science. A lot of this information is on there with videos and information about the complement system and the different kidney conditions that the complement system can impact. So with that said, thank you very much for your attention.